Good morning, everyone. I'm Kay Hoare, Director of the UK Squirrel Accord, and I wish you a very happy Plant Health Week. This week is especially dedicated to raising awareness of the importance of having healthy plants in our lives. Not only does around 80% of the food we eat come from plants, we're also supported by trees providing essential services like creating oxygen, cleaning air and water, and providing timber. Alongside human benefits, our trees and woods provide vital homes for wildlife such as red squirrels and many other mammal, bird, insect, and plant species. Across the UK, there are significant tree planting and woodland creation targets and actions underway. However, there are also serious threats from tree pests and diseases, such as Clara, dieback of ash, and acute oak decline. An often lesser known threat comes from grey squirrels. These introduced mammals actively bark strip young broadleaf trees, stressing, weakening, and in too many cases, killing ecologically and economically important species. Grey squirrels may also act as a vector for pathogens to infect trees through the open wounds they create. Oak trees are a particular target for bark stripping, and the loss of these trees or a reluctance to plant them because they will be damaged can have knock-on impacts on the 2,300 species that oaks support. Our trees and woods are under great pressure at the moment, and we need to do everything we can to ensure they have a healthier future. The UK Squirrel Accord and supporters are funding research to create a grey squirrel oral contraceptive and species-specific feeding hopper that, in the future, could be used alongside or instead of traditional wildlife management methods. Fertility control is increasingly being used around the world as a solution to tackle problematic species, and we need to explore new advancements in science to solve old problems. For today's webinar, I'm joined by three members of the fantastic Animal and Plant Health Agency team currently conducting the research into grey squirrel fertility control to protect tree health and red squirrel survival. Rebecca Pinkham is the project lead and is working on the oral contraceptive. Sarah Beatham leads the research on the species-specific feeding hopper, and Simon Croft is focused on the DEFRA-funded modelling. This work has successfully reached the end of the initial research project, and we are celebrating its progress. Research will continue while a review of the work is carried out so we can effectively identify essential next steps, and we will create a new fundraising campaign to enable research to continue and to deliver landscape scale trials currently being developed. A big thank you to everyone that has supported and collaborated on this work so far. We are super grateful to you all. So now we will have a presentation from each of the team on their area of work and then follow up with a Q&A session at the end. If you have any questions you wish to ask, please put them in the Q&A section of Zoom to enable us to keep track of them. And do use the chat function to say hello, share any news and events around Plant Health Week, and for any general discussions around fertility control, grey squirrel tree damage, or anything else. So our first speaker is Rebecca Pinkham, and I will hand the screen sharing, over to you.
Hi, thanks very much. Just get the screen going there.
Oh, apparently it's also David Attenborough's 98th birthday, so happy birthday to him.
Is that all up for you?
Yes, that's working fine.
Cool. So yeah, I'm going to give you an update on the lab side of the project. So overall, we are developing and delivering oral contraceptives for the grey squirrel. Now, for those of you that haven't been to any of our talks before, the project is made up of 3 tracks. Now, the first being the pursuit of a suitable oral contraceptive for grey squirrels and involves the actual development in the lab. The second, and equally important, is a track looking into the delivery of a contraceptive so that when we have a suitable one, we know that we can deliver it to squirrels, only squirrels, and to enough squirrels that we know it will have the required effect. And the third part is modelling, where we would field data alongside computer modelling to predict the impacts of population control methods on a landscape and can use this to compare the relative effort needed to achieve population reduction in grey squirrels. Now, I'm going to take you through the progress that we've made throughout the whole project on the development track, and we'll hand over to my colleagues in a minute for an update on the other 2. So for development of a contraceptive, we need to choose an appropriate target in the reproductive system. In mammals, this is primarily controlled by the hypothalamus, which monitors and causes the release of hormones for reproduction. When required, the hypothalamus produces gonadotropin-releasing hormone, GnRH, for the anterior pituitary, which oversees all sex hormones and stimulates a cascade of reproductive hormones in both males and females. By disrupting all or part of this process, we can cause infertility in both sexes. And by preventing the action of one hormone at the top, we can prevent the cascade of all reproductive hormones, essentially shutting down the whole reproductive system. Alternatively, we could target lower down in the production of more specific reproductive hormones, such as testosterone or the female reproductive hormones. Now, although there are a number of contraceptives available in general, the pursuit of a contraceptive that's suitable for wildlife isn't an easy task. It needs to be remotely delivered, so ideally edible, long lasting, so an animal doesn't have to eat it every day, and have a low likelihood of any side effects. One key solution is a contraceptive vaccine that causes the body to produce antibodies against reproductive hormones and reduces their activity, preventing breeding. There are injectable vaccines, but moving to an oral version has different challenges, particularly from the acidic stomach environment, and this is why we don't see many oral vaccines in general, even for humans. Now, it's hard to sum up all of the research that we've done over the last few years in one slide, but I'm going to give it a go. Throughout the whole of the current project, we've been working with a number of collaborators to identify, develop, and refine a range of oral contraceptive candidates that have included both vaccine-based contraceptives and chemical contraceptives. We've had the most success using a GnRH-based immunocontraceptive vaccine. With this, we need to test for the strength and duration of an immune response. So through multiple lab tests over the years, we've worked to identify some key components that act to make the vaccine as potent as possible and give us the highest chance of causing infertility. Using rats as a model species, we've been testing multiple variations of the vaccine formulations and measuring their effect through the production of antibodies and through their ability to prevent breeding. In parallel to this, one of our focuses has also been the development of a captive breeding colony of squirrels that we could use to test a contraceptive on when we've reached the final recipe, as it's crucial to have a breeding baseline to compare a treatment effect to. Throughout the testing phases, we've been investigating different dose levels to try to achieve a strong effect in the majority of treated animals and comparing different dose schedules informed by our fieldwork and individual bait uptake patterns to try to develop an effective dosing regime for the practical field application. Now, giving a guaranteed vaccine dose through a syringe is very different to it being eaten from a feeding hopper in the field. So we've also been working to test all sorts of different bait type combinations to make sure that we'll be able to deliver the vaccine in something that will be attractive and effective for animals in the wild. After trying out successful combinations of all the above, we've started to test oral doses of the contraceptive vaccine in captive squirrels to test the strength and duration of the effect in our target species. So about the contraceptive itself, in partnership with local and international collaborators in the last couple of years, out of all the candidates, we've been focusing on the development and refinement of the immunocontraceptive vaccine formulation that targets GnRH, which was at the top of the hormone cascade. This means it should work in both males and females. Using rats as a model species, the most successful candidate has been a recombinant protein vaccine developed by a company in France. This is added to an adjuvant derived from natural sources to boost the immune system response. Importantly, our most effective formulation combines this with a novel protection and delivery technology SPECS, which is patented and developed by Spiromex Limited in the UK. These are the spiky capsules that you see pictured here. Environmentally friendly plant-based microcapsules consisting of empty pollen spores that are designed to protect the protein through the digestive system and improve uptake by the immune system. And we've shown this to significantly increase the potency of the oral vaccine itself. Now, we're also designing ways to deliver this vaccine through a bait that is palatable and attractive to squirrels, but importantly can be delivered in a liquid or a semi-solid state so that when we move to field delivery, it's not something that squirrels will be able to take away in cache. This prevents the risk of the vaccine getting into the environment outside of our specially designed feeding hoppers. And now to the highlights so far. So with this vaccine, we've made some significant progress in the last couple of years. We've achieved strong immune responses through syringe feeding in rats. And we've also achieved infertility through a feeding dose where the vaccine was combined into a sweet liquid bait for rats to eat. This is, as far as we're aware, the first time any group has managed to get a functional response from an immunocontraceptive vaccine that's been delivered in a voluntary bait. Now, the next point might sound a bit counterproductive, but we've managed to achieve some really great breeding success in our breeding colony. These were a group that hadn't been anywhere near a contraceptive. And this is really important because reproductive research relies heavily on being able to have a baseline for comparison to a treatment. It'd be impossible to tell if a contraceptive were working properly if no control animals were breeding either. And we haven't tested it in the breeding colony yet, but we have taken a major step forward into squirrels in the last year and have made important achievement of successfully producing an immune response to our vaccine through oral delivery in squirrels. This is a big movement forward in the project. It's the first time we've proven an oral response in our target species and indicates that the vaccine can be used in squirrels. We just need to keep working on improving the consistency and the scale of responses and the development of a suitable bait format to get the vaccine delivered to them in the right way and at the right quantity. Now, although our main focus, not our main focus, there's an ongoing target throughout the project to maintain the actively breeding colony. As I mentioned before, this comes from the need for a stock of squirrels of known breeding history and active breeding status for use in a final proof of concept study where we can prove the link between treatment and infertility. Contrary to expectation, this has been one of our challenges, with captive grey squirrels seemingly reluctant to breed in captivity. However, we've recently overhauled our housing layout, and the levels of breeding success seen in spring and summer 2023 have significantly increased from previous years. Our colony has consisted of around 35 to 40 females, and after a big change in the housing structure, we've managed to achieve the proportions of females breeding last year equivalent to what we see in wild populations. And this confirms that our colony can be used to test a contraceptive effectiveness when we have a vaccine bait ready. So what's next? We need to work on developing a vaccine formulation further to make it more effective in a free feeding format. And we need to work on defining the most suitable bait and dosing schedule to give us an effective, stable, and practical bait that will be suitable for larger scale use in the field. We'll be looking at how to translate the field data on wild squirrel behaviours into the vaccine makeup, where patterns over time inform how we need the vaccine to perform, such as the total visits per individual over time, leading into testing practical dose delivery schedules. How often they return to feed will heavily guide the number of doses we can effectively deliver and gives us our testing range. We will need to then ensure that the chosen vaccine format produces the results that we want over the dosing scale that we know we can achieve in the field. In order to proceed to a test in the breeding colony, we need to identify a formulation that produces a high level of titers in squirrels via free feeding and can be sustained for at least the length of the breeding season. To obtain this from where we currently are, additional pilot trials will be necessary to test a free feeding vaccine in squirrels and test various field suitable bait options. We hope to be using our breeding colony to test from 2025 onwards, provided the next year of research is successful. We also want to start looking even further forward. We're working at a very small testing scale at the moment, so we'll be looking into how we can scale up the construction of the vaccine and its components, including the environmentally friendly processes for the extraction of the pollen spores. We also want to begin to look into potential manufacturers for the total product and define the next steps that will be needed to get the vaccine that we have to a point that it can be taken through the registration process necessary to become a usable product. With the UK Squirrel Accord, we've mapped this out as the 4 phases needed for delivering fertility control as a practical tool in the squirrel control effort. Now, we're currently in the research and development phase, which will hopefully give us a suitable oral contraceptive, the delivery and deployment strategy, and a model that allows us to design coordinated control. Next comes the landscape phase that is currently being planned. Here we'll use a proof of concept from the research phase and move into field trials, both on the small scale to test the contraceptive itself on isolated populations when the contraceptive is available, but also on large scale studies looking into deployment itself, evaluating the feasibility and effort required for practical delivery at a landscape scale. These effort studies could be conducted prior to finalisation of the contraceptive itself, provided that the bait format is known. The third phase is the registration phase. We're currently working with a company specialised in taking veterinary products through registration to advise us on the likely data package that will be needed for a product of this type. As soon as the vaccine formulation itself has been finalised, we would hope to progress and carry out field trials under a research licence. We can also move forward and engage with suitable manufacturers and work with the registration authorities to put together the registration package itself. This will include a lot of testing for safety and environmental impact. Finally, we get to rollout, where there will be large-scale manufacture and availability for practitioners. Throughout this rollout phase, we'll still be looking at monitoring the efficacy of widespread delivery and developing best practice guidance from on-the-ground experience. From where we are now, we believe that with continued effort and funding, progression through these phases and a contraceptive rollout could be achievable within the decade. And last but not least, I just want to say a huge thank you not only to the Squirrel Accord for their support of the research, but to all of our colleagues and collaborators who are working so hard to make fertility control for squirrels a reality. Thank you very much.
That's brilliant. Thanks very much, Bex, and great to see the progress on the oral contraceptive side. And now we'll pass over to Sarah.
Right. Sorry, just get rid of this. Right, there we are.
Yeah, that looks good.
Okay, good. Right. So thank you to everyone for joining us for this webinar. So I'm going to present the research we've made so far on delivering oral contraceptives to grey squirrels. Okay, so this research is focused around 3 key study questions. So the first one is, how much contraceptive bait would a grey squirrel eat? So this tells us the potential contraceptive doses we could achievably deliver to grey squirrels in woodlands. Second, how many grey squirrels would eat a contraceptive bait? So this provides us with a measure of potential contraceptive effectiveness. And finally, can we prevent other animals, including red squirrels, from eating a contraceptive bait? So as Bex mentioned before, the contraceptive vaccines we're looking at could potentially affect other mammals. So we want to make sure that we're feeding these contraceptives to grey squirrels only. So to measure how much contraceptive bait an individual squirrel would eat, we microchipped 202 squirrels in 6 woodlands. So that was between 21 and 44 squirrels per wood. So we released them into the woodlands, and then we deployed our feeding hoppers. So these were deployed in 2 woodlands in winter, 2 woodlands in summer, and 2 woodlands in spring. So as you can see from the red location points, they were evenly spread throughout each wood. And to simulate the deployment of a contraceptive, we pre-baited them for 1 week with hazelnut paste with the doors propped open to allow the squirrels to get used to using them. And then we closed the doors and we baited and monitored them for 4 days. So this picture on the right shows the internal structure of our bait hoppers. So this was developed by the company Nature Counters. So it comprises of a bait compartment, which has to be accessed via a weighted door. So as you can see, the squirrel will demonstrate it by using it. The squirrel has to push open the door, and there's a microchip reader at the front of the door that records the date and time of each visit by a microchip squirrel. And then in summer and spring, we added a weighing platform to weigh the amount of bait taken per visit by each squirrel. So within the 4 days that we monitored in the 6 woods, 125 microchip squirrels, that's 66 females and 59 males, made 1,676 visits to 111 different hoppers. So this is obviously a lot of data to analyse, but we summarised the amount of bait taken by each squirrel from hoppers in the graph presented. So you can see the amount of bait, the weight of bait taken along the side, the number of squirrels that were recorded along the top, and these are split into males in blue and females in orange. So on average, 47 grams of bait was taken from hoppers by females and 39 grams by males in the 4 days of deployment. But as you can see, we had much better bait uptake in spring with fewer hoppers. So there was a lower hopper density, meaning there was fewer number of hours required in the field to deliver bait to the hoppers. And also we had better bait uptake by females in spring. So we specifically, for contraceptive to be the most effective, we want to target ideally the breeding females in the population. And spring is a very good time to deliver contraceptive, as it's immediately prior to the second main peak in breeding in grey squirrels. So this graph shows the relationship between the time squirrels spent visiting hoppers based on the microchip records versus the amount of bait taken per visit. So as you can see, there's a very strong relationship between these 2 factors, with overall 1 gram of bait taken by each squirrel per minute. So when the squirrels were feeding from hoppers, they typically went in and out of the hoppers quite rapidly and frequently. So you didn't typically get squirrels that sat inside hoppers and fed for a long time. So that's good for delivering a contraceptive, as it means that individual grey squirrels are not going to dominate the hoppers and prevent other individuals from accessing them. So as the time spent visiting hoppers could be related to the amount of bait taken, we then could compare our seasonal results across winter, summer and spring. So as you can see, we had the best bait uptake in winter and spring. And females on average made 12 visits to 3 hoppers within the 4 days, compared with males making 15 visits to 5 hoppers. So although males made slightly more feeding visits to hoppers than females, this demonstrates that they certainly weren't dominating hoppers, so we could still target our females effectively with a contraceptive. And again, with the average visit of just 39 minutes, this showed that we didn't have squirrels dominating hoppers. Now, part of the reason for this is because the bait we used was like a thick liquid, so it was a hazelnut paste. And that meant individuals couldn't easily remove it from the hoppers and deposit it around the woodlands. And that means it could be contained and other wildlife species couldn't access a bait like that. So we used the data to model the probability of female and male squirrels entering hoppers by season. And we also related this to the number of squirrels per hopper in each wood. So as you can see, squirrels were more likely to enter hoppers in winter, but specifically females were more likely to enter them in spring. So this might be the best time of year to deliver a contraceptive. As I mentioned before, it's immediately prior to a main breeding, peak in breeding. But also, if the number of hoppers per squirrel was at least one, if there's at least one hopper per 2 squirrels, I should say, then the probability of a female entering a hopper was over 80%. So to ensure we had enough hoppers per squirrel, you can estimate the initial squirrel density using cameras. So we developed a camera method and published it, where you put out cameras in woodlands for 5 days. You can estimate the squirrel density. So then you can ensure you can put the minimum number of hoppers out to minimize the amount of time spent visiting them in the field, but ensure that there's enough squirrels, hoppers for the squirrels in that woodland. So how many squirrels would feed from a contraceptive bait? So in order to answer this question, we deployed our hoppers in 14 woods, and these were distributed around Yorkshire and in North Wales. So again, we pre-baited them for 1 week with hazelnut paste, and then for 4 days, we added a pink dye to the bait and monitored them. So the dye, when consumed by the squirrels, turns their hair pink under UV light. So by trapping them at the end and taking a hair sample, we can see the proportion of the population that's taken a bait from hoppers. So this graph summarizes the data we've collected from these 14 woods. So it shows the percentage of squirrels in each wood that managed to eat bait from the hoppers. And this is related to the squirrel density that we measured in each wood. So you can see the pale blue is a low squirrel density up to a high squirrel density, which is shown by the dark blue. So as you can see, when squirrel densities are low, we get very good bait uptake results. So with 3 hoppers per hectare in summer, we managed to deliver bait to over 70% of the squirrels in 3 woodlands. When the hopper density is lowered to 1 per hectare, typically you don't have enough hoppers per squirrel to ensure that over half the population feeds from them, unless the squirrel density is very low. So in summer 2022, we also trialled a longer bait deployment, so 12 days versus 4. So we wanted to see if that would massively improve the percentage of squirrels eating bait from hoppers. And as you can see, the results were actually fairly consistent with other woods done at different times of year and with similar hopper densities. Also in 2023, we decided to test an 8-day deployment, and we were looking at varying the hopper density from 2 to 3 per hectare. But again, as long as the squirrel density isn't too high, the results are fairly consistent. So on average, we were getting between 55% and 65% of the squirrel populations across all woods taking bait from hoppers. So from this, we could conclude that most squirrels in most woods would feed from bait hoppers, regardless of season and hopper density. So our final question was, can we prevent other animals from eating a contraceptive bait? So as you can see, when we conducted all of these hopper trials, we monitored the hoppers with cameras to see who else was trying to use the hoppers. And we photographed a range of wildlife species around the hoppers. So overall, we monitored 322 hoppers in 14 woods for those same 4 trial days. So the hoppers recorded every time the door was opened, and then we could match this to the footage on the camera to see who opened it. And from this, we recorded around 180,000 photos or videos of animals around the hoppers. And of these, 5,000 visits were made by squirrels where they were able to open the hopper door versus 4 visits by mice. So mice were the only other animal that actually entered the hoppers and were able to open the door. But we can't be certain if they actually managed to feed from the hoppers. But from these results, we concluded that in most areas, a contraceptive bait could be delivered to a large number of grey squirrels with minimal impact on other wildlife. However, what about red squirrels? So in most areas in the UK that have grey squirrels, there aren't red squirrels present. But if you want to deliver a contraceptive bait in areas where both species are present, then you need a more sophisticated tool to select between the 2 species. So after trialling a few different solutions, we decided to use body weight as the selective tool. So these automatic weighers, designed and built by a red squirrel practitioner, Barry Bickerton, were deployed in various woods in Cumbria, Conwy, Northumberland, and North Yorkshire. So we're trying to gather as much red squirrel body weight data as possible. And the weighers were typically baited for 1 to 2 weeks, and we monitored them with cameras, so when they recorded a weight, we could see who was being weighed. So this table summarises the maximum red squirrel weights we achieved from each area. So between 2021 and 2022, we didn't obtain any weights of red squirrels of over 430 grams. So in 2023, we put the weighers out in North Yorkshire at a time of year where we would expect the red squirrel females to be pregnant. And we then obtained slightly higher maximum red squirrel weight of 435 grams. Later that year, we then deployed the weighers in areas where there was both red and grey squirrels present to see if that had an effect on body weight. And these weights were slightly higher still at around 438 grams. So the vast majority of weights recorded for red squirrels were actually below 400 grams. There was only a few that were higher than this. And this may be because there was a few heavier individuals in the populations, maybe pregnant females. But also this could be just simply because certain individuals were applying extra pressure onto the weighing platforms from the way they were standing on them. But to be certain and to be conservative, we decided that if you chose a minimum weight threshold of 450 grams to open a door, then that should exclude all red squirrels from a bait. But to be sure, we had to then compare it with the grey squirrel body weights to make sure that this would be effective and still allow enough grey squirrels to access the feeders. So first we took our body weights from the microchip trials that we conducted at APHA. So that was 205 squirrels. And between 92 and 96% of them were at least 450 grams. Then we widened the analysis to include a lot of weight data that was gathered by grey squirrel practitioners. So this was from multiple areas across Northumberland and Scotland, and from lots of different times of year. So from these 1,692 body weights, we saw that between 76 and 88% were at least 450 grams. And most of the individuals that were below 450 grams, from our analysis, we could tell that they were typically juveniles or subadults, and therefore non-breeding and not the target of our contraceptive. So from this research, we concluded that a weight threshold of 450 grams could exclude all red squirrels while allowing at least 90% of adult greys and at least 76% of juvenile and adult greys access to a contraceptive bait. So then we decided to test this theory by developing and testing a prototype grey squirrel feeder, again developed by Barry Bickerton. So he put this feeder out for 3 weeks and baited it in a woodland in Cumbria where we knew there were both red and grey squirrels present. So the feeder has a motion sensor, and when it detects movement on the weighing platform takes the weight of whoever's on it. And if that weight is 450 grams or above, then an electronic door is opened and the bait can be accessed. So grey squirrels tried to access the bait on 13 occasions in the 3 weeks. And as you can see, on 7 of those occasions, they were successful. For the other 6 occasions, this could be because the grey squirrels' weight were lower than 450, or it could be because we need to work a little bit more on the sensitivity of the weighing plate. But this was certainly a good start. So red squirrels tried to attempt to access the feeder twice, and on both times they failed. And as you can see, they were certainly persistent and they gave it a good go, but they were unable to access the bait. So going forward, as we've proved the concept of this feeder, we are currently consulting with manufacturers as to how we could produce a more robust field feeder that could be manufactured cost effectively. And we're also looking at applying the technology this feeder is based on into developing a selective cage trap for grey squirrels that could be used in areas where there's both reds and greys present. So back to our key study questions. So in terms of how much contraceptive bait would a grey squirrel eat? So from our research, we can say that squirrels will typically make multiple visits to hoppers on most days of a bait deployment. And they will probably eat around 10 grams of bait per day. And in spring, they will tend to eat even more than that. To answer how many grey squirrels would eat a contraceptive bait, our research suggests that most squirrels in most woods would eat a contraceptive, as long as there are enough hoppers per squirrel. And finally, can we prevent other animals, including red squirrels, from eating a contraceptive bait? So our research suggests that it should be possible to feed a contraceptive to large numbers of grey squirrels with negligible impact on other wildlife species, including red squirrels. So going forward, we're collaborating with Northumberland Wildlife Trust and partners to produce a bid for the Red Squirrel Recovery Network project. So this is a Lottery Heritage Fund project. And as part of that, we're hoping to conduct landscape level trials with volunteer groups where we can measure bait uptake on a much larger scale and also assess the species specificity of our feeders to a greater extent. So I'd just like to thank our funders, the UK Squirrel Accord and DEFRA, all of our collaborators, and I'd particularly like to thank the landowners, practitioners and volunteers that have contributed all of their land, time and data to our study. Thank you all for listening as well.
That's great. Thanks very much, Sarah. There's so many people involved, isn't there? It's fantastic. That's brilliant. Simon, can I hand over to you to look at the modelling? Brilliant.
In theory, if I can manage to work out how to use my system. Right. Can you see my presentation?
I can indeed. It looks good.
Okay, great. So yeah, I'll kick off straight away. As Bex mentioned, so I'll be talking about the modelling aspects, the third strand of this work, which has been funded by DEFRA, mainly looking at the impacts at a population level if we were to have an oral contraceptive available, and comparing those to more traditional methods for population control. So why do we really want a model? Well, there's three main reasons really. The first is it helps to provide a structure for us to gather together and combine all of the existing pieces of information that we already know on the ecology and on existing methods for population control into one place, and hopefully then to be able to identify the key gaps that we need to fill in order to make robust predictions. Once we've done that, we can take this model and based on the assumptions that we have in it, start to look at what might happen at a much larger scale than would be possible to look at empirically, to go out and look at in the field. So we can start to look at future strategies for how the population control might work with different interventions, different types of control in order to, I don't know why that's moved on, sorry, in order to plot a way forward. And then the third aspect really is while the fertility control research is going on, while the contraceptive development is continuing, we're then able to inform and hopefully feed back into that work from what we find from our models. So for instance, looking at whether there is a minimum target for contraceptive to be effective and under what circumstances it might be in order to help with the development. So just a bit of information about the model. So it's what's known as a spatially explicit individual-based model, which sounds very complicated, but unpacking that, really what that means is that we try to represent an abstraction of the landscape, so try to represent the habitat in which squirrels live and move around in. And then into that landscape, we pop in our squirrels and allow them to move around. Obviously, we simulate natural mortality and then reproduction as well. And then within that landscape, we are able, as a separate layer, to add in methods for population control and allow the squirrels to interact with those as they go along. So really, I just wanted to present quickly a brief recap of the initial results, a bit of a history of where we've been with the model and what we've done. So initially, based on very early field work from Sarah, looking at the efficacy of contraceptive or bait uptake in the feeders with a fixed density of feeders, we wanted to be able to compare against culling using live traps in order to try and work out how the two might potentially compare and maybe suggest a level at which we would need to achieve contraception in order for it to be in order to potentially replace live trapping with contraception for a similar amount of effort. And this initial attempt was obviously based on a number of assumptions, a number of estimates. Firstly, looking at the likely interaction or the rate at which squirrels are likely to interact with live traps in the environment, which we took from the literature. And then, like I say, looking at the early data from Sarah's studies. And this suggested that we would need to potentially need to contracept 60% of squirrels in order to achieve eradication with an effort comparable to culling. But again, this was based on the idea that it would take less effort, so less effort to maintain feeders than it would traps. So based on the idea that traps need to be visited once a day, whereas feeders may only need to be visited to top up the bait every 5 days. Obviously, like I say, these were based on initial assumptions and initial data. And as Sarah's work has continued, we've had more information available. Our aim was to be able to look at different deployment strategies, so different densities of feeders in the environment and different densities of traps in the environment, and see how population control would change depending on these different scenarios. So the way in which we did that, firstly, we were able to obtain a better estimate of squirrels' interaction with live traps through Sarah's early work. And then on the right, this is an early analysis of the work that Sarah was showing, looking at bait uptake, the proportion of individuals in a population that were found to be feeding from the feeders. And this shows, as Sarah mentioned, that the overall proportion that are able to be targeted is related to the ratio of squirrels per feeder. Obviously, in going down, so the more squirrels we have per feeder, then the lower the proportion that are able to be targeted overall. And we applied this into the model then to be able to vary, like I say, the densities of deployment and produce another comparison. So with these updated numbers, we started off by looking at varying live trapping. And what we found was that actually we could achieve eradication with a lower density than we previously attempted. And with similar trapping efficacy efforts to the large-scale removal on Anglesey, we achieved eradication in similar time. So I think we assumed about half a trap per hectare over I think 20-day period. And that achieved eradication within 10 to 15 years, which is similar or along the same lines as Anglesey. However, when we then deployed contraception at the same densities in order to provide us the comparison again in terms of effort, we found that the contraceptive delivery didn't reach the 60% target that we were looking for in order to achieve eradication with similar efforts. And this is because of the relationship on the right that at that density of feeders, the number of squirrels per feeder increased to a level where we weren't able to deliver the contraceptive successfully enough to be able to affect that eradication. So we moved on instead to look at what happened if we initially trapped, so 1 year of trapping and then performed contraception. And what we found is by lowering then this starting density of squirrels, we were able to achieve eradication again using fertility control, and actually managed to achieve that in similar time to trapping alone. But obviously, assuming that the feeders needed to be visited less than the traps in equal number, equal densities, and in a completely equal setup, then that could mean that we could actually achieve eradication in this manner with potentially less effort. So what were the next steps we moved on to? So it was to look at this a little bit further and to try and refine our estimates for effort to provide a more direct comparison. So along the idea that with lower frequency, with less frequent maintenance, it means that we could potentially put out more feeders, but still achieve eradication with fertility control alone, for instance, but using either less or similar effort to culling. So this has really been the subject of our latest published work. Using our annual model, so as I've said, continuing to revise estimates for squirrel numbers, our management efficacy, and then the labour attached to those. We looked at a much wider suite of potential strategies for both live trapping and for contraception and delivery of contraception. So we found that if we were using live trapping everywhere, as we've seen throughout, this achieves eradication quickly, so 4 to 5 years, within a population similar size to Anglesey, and that the optimum annual effort is around 11.25 trap days, which interestingly enough corresponds to roughly a trapping density of somewhere between 1 trap per 2 to 4 hectares, trapping 5 days per month for the 6 months from March to September, which I think is the recommendations that you'll find in the Grey Squirrel Handbook from John Gunnell and Harry Pepper, sorry, which is interesting, but suggests that actually the current recommendations are potentially quite close to the optimum that you could do for live trapping. We found that eradication using contraception can be achieved, and obviously this is with lower annual effort than culling, about a third of the effort of culling, just in terms of maintenance of devices, but is much slower, and at best can be as quick as 23 years, but equally can take up to 40 years to eradicate the same population as it took trapping 4 to 5 years to eradicate. Again, we looked at a combination of culling followed by contraception, and this did still reduce the time to eradication substantially, so knocking off 7 to 12 years from our estimates. But again, not sufficiently to make it as much effort, like as little effort as trapping everywhere. However, up to now, so looking at eradication as a target point has favoured culling from the perspective that it's an intense effort, but then once you've eradicated the population, you can stop. We wanted to look at what might be achieved if we were looking instead at population suppression. So looking at ongoing control, in which, of course, contraception, fertility control with lower effort could potentially provide an excellent solution. So for population suppression, post-cull, what we were able to show is that if we could contracept 50% of squirrels, then we would be able to maintain low populations using fertility control. However, for similar effort, we would need a 90% effective contraceptive. But I should caveat this by saying that this is probably an upper estimate. And I'll draw your attention to earlier in the slides where I point out that in our estimates for labour in this study, we didn't account for travel time, which may be similar between the maintaining the feeders and live traps, but certainly we didn't account for the time taken and the labour involved with dispatch, which obviously you wouldn't have with maintenance of the feeders, and could increase the amount of labour considerably. So it's worth saying that this 90% effective contraceptive for similar effort between culling and fertility control is likely to be lower than this. For the first time, we have moved on also in this study to looking at what the impact of partial coverage may be. So up until that point, we had only considered what happens if control is applied everywhere. But realistically, there are going to be gaps in the landscape, and it's going to be quite difficult. Completely coordinated effort is going to be difficult to achieve. In order to do this, we looked at the previous studies that have been done surveying public attitudes towards different control mechanisms, different control methods, and applied these to landowners, as if assuming that they might follow the same patterns. So that implied that you would achieve, I think, less less landowners accepting culling, around 40% of landowners, whereas around 64% would accept non-lethal control like contraception. It's important to say that with neither method, complete eradication was no longer possible. So we still maintain pockets where squirrels were not being controlled, populations were not being reduced. So obviously we couldn't completely eradicate from the landscape. But what we found is that contraception could reduce numbers more than culling at landscape level with less effort, which was very interesting. I think so that was assuming a perfect contraceptive, to reduce numbers by a similar amount. We were able to do that with only 75% effective contraceptive. Quite interestingly, the two methods produced completely different footprints. So with culling, obviously looking at less coverage but more intense removal, versus contraception, which was more widespread and less intense removal. And so actually our conclusions from that is that they may be applicable to different targets. So for instance, where we want to reintroduce red squirrels, for instance, we're looking at having to almost completely remove grey squirrels. So this is where culling using live trapping may be the best option. Whereas for fertility, for contraception, if we're looking to alleviate tree damage, which may involve just reducing populations slightly to remove some of the stress on the populations, then that may be the best approach, as we can target more woodlands with it. So where are we going next? So as I think we've now decided we've run our course with our annual updating model, and what we need to do is to provide more granularity, so we can start to look at how management actually is implemented on a finer scale. So we are slowly moving our model towards a weekly updating step, which means that we can now simulate any routine within a 20-hectare block of wood on a weekly basis. So for instance, we can have a rolling program of trapping or of contraceptive deployment, moving around an area, and see how the populations potentially respond to that, as opposed to considering all of our control happens at exactly the same time. And we're also looking to include interannual tree masting. So the levels of masts have a massive impact on squirrel populations. So in a good year, you can have huge bursts in population, whereas potentially if you have a poor year and you target culling or target removals in those years, then there may be a benefit, a much better impact in that sense. So the results on the right just give you an indication
Sorry, Simon, we've lost your video feed for your presentation.
Oh, does it come back?
No.
Okay, just give me 1 second.
Yeah, sorry.
Capture background is taking longer than expected.
I really wanted to find out what the graphs meant on the right-hand side, that's why.
As we'll get there. Is that better?
Yes, great, thank you.
Yeah, okay, sorry. Poor Wi-Fi connection, I think must be the case. So these are just an illustration, and it's worth noting that I am aware that some developments that I need to work on and need to implement in the model will change this picture. But it's just to give you an idea of what we're looking at the moment. So this is looking at a scenario where we are trying to rid an area, location of grey squirrels, and then provide continuous protection. So it's along the lines that we were looking at before of how much, what level of coordinated effort do I need in order to achieve my goal? And so it's looking at, firstly, with culling, looking at a buffered ring around that area to see what the probability is of me keeping my area, my location of interest free of squirrels over time. So initially looking at a radius of 3 kilometers, 5 kilometers and 10 kilometers, culling in all woodlands around for 10 years. And then what happens if I stop after 10 years, and continuing to monitor the population recovery for a further 10 years. I mean, it's quite interesting. What we see is that potentially we need a ring of at least 5 kilometers in order to maintain population grey squirrel freedom for while we are controlling the population. But actually, if we want to maintain that even once we stop, then for a period of time at least, then we're potentially looking to have to double that distance to 10 kilometers. And this is really just about looking at the dispersal and movement of the long distance dispersal and movement of grey squirrels in our model. We're going on as well at the same time to then say, well, what happens if, for instance, we have a 5 kilometer radius where we're culling in the center, but we replace the outer 5 kilometer radius with contraception, lower effort contraception instead. And what we can see is that actually by doing that, we managed to produce the same results as if we were trapping in the whole of that 10 kilometer radius, which is good news. And it makes sense from the point of view that it's in the outer 5 kilometers, the job that it's doing is to reduce the amount of pressure on the grey squirrel populations, and hopefully to reduce the amount of, to provide space for squirrels to move between woodlands and not to have to continue to move and seek the space we're creating towards the center of the area, our area of interest. In terms of where we want to take this, so there's a couple of further aspects, one of which is to try and look at practical advice that can be implemented in the field by practitioners. So we'd like to use the model to try and identify particular woodland characteristics, which perhaps where there's limited resource might indicate or might give an indication of priority. So if I can only work in 1 or 2 woodlands or provide a certain amount of effort, where should I target that? And the idea behind trying to identify the woodland characteristics is that we then don't necessarily need to run the model every time, but it's possible to use other resources to evaluate that for different locations quite quickly. And then, as we've I think alluded to, and Sarah's mentioned, I think in the work that she's looking to do, we are looking at implementing potentially other existing methods to compare against. So for a number of years, we've been looking at trying to gather the data to be able to include shooting as a comparison, as another common method for controlling grey squirrels. We're also gathering data on good nature traps, and hope to be able to include those again as another comparison point in the model. And at some point, although others have looked at this in more detail, and I think we can probably rely on their outputs, so it might not be necessary, but at some point, as part of an integrated approach, we may also include pine marten as part of the toolkit for controlling grey squirrels, and see how that might integrate in with all of the other available methods. And that's me.
That's brilliant. Thanks very much, Simon. It's really important to have this modelling work done so that we can understand the potentials for the future. We've got a few questions that I can put to our speakers. I might start with Bex around the contraceptive work. So Bex, is the contraceptive effective on both male and female? If she's there.
Short answer, yes. So the contraceptive we're developing targets GnRH, which is at the top of the hormone cascade for both male and females. So as long as we can get it into both of them, it should be effective on both males and females, yes.
Brilliant. And how often do you envisage a squirrel, male or female, to have to eat the dosed bait to remain infertile?
So we're still working on that one. At the moment, we're running a schedule of between 4 and 6 doses over 1 month. We would hope to reduce that, but that's one of the parts that we're working on. And we hope with kind of this schedule that would give us an effect for at least covering 1 whole breeding season, but we would hope that we can get it to a point that it would be at least a year or more. So the contraceptive, if you want to get it into the population, it would be either once or twice a year that you have to deliver it.
So putting it out in the field for a month, and then that would last for at least a breeding season or maybe a year.
Yeah, that's what we hope. Yeah.
Yeah, brilliant. And would the contraceptive affect other species feeding on grey squirrels?
Yes, get quite a lot of questions on that one. So the hormone that we're targeting is a mammal-specific hormone, so there wouldn't be any risks at all to any birds either predating on squirrels or birds that would kind of accidentally get a hold of the contraceptive, although that shouldn't happen. As well, because it's a vaccine in a bait, when it's digested, all that's left in the animal is antibodies, and that shouldn't transfer down the food cascade, down the trophic levels, so it shouldn't affect it. But that's something that we're also going to be looking at through the registration testing. There's going to be a lot of safety and efficacy tests that we need to carry out, so that will be something that we are wanting to prove. We know that it shouldn't, but we'll be trying to prove that as well.
Brilliant. Thanks very much, Bex. And a few for Sarah. Bettina wonders if unmicrochipped squirrels also took advantage of the feeders, and if you knew that.
Yes, that's what we use the rhodamine bait dye for. So for those studies, because typically we went into a wood and tried to trap and microchip as many squirrels as possible. But obviously when you're trapping, you tend to then start recapturing the same ones. So we typically caught around 2/3 of the population each time in each wood, we believe, based on capture numbers and population estimates. But with the rhodamine, we could basically try and catch all of the squirrels at the end and then test them to see who had accessed the bait. And that gave us a much bigger picture in terms of the percentage of population that would actually take bait from a feeder. And when we did that, we got higher levels of bait uptake of over 70% in certain circumstances. Yeah.
Brilliant. Thanks, Sarah. This is a question, I think, about some of the graphs that you had up and whether those durations you're showing were really in minutes or seconds. I think that was it for how long grey squirrels were visiting a feeder?
Yes, they were actually minutes. So that was total number of minutes over the course of 4 days, though. So, you know, that they were typically visiting for up to 39 minutes at a time. Now, the way I defined a visit was just when the squirrel was literally at the entrance of the feeder and then moving in and out. So obviously they weren't in the feeder for the full amount of time. which is why they were only taking about 1 gram per minute. So the reason I did that was because actually they were going in and out of the feeders. We were getting lots and lots and lots of microchip records. So I had to define a visit in some way. So it was all the consecutive microchip records within 2 minutes. And that's how I defined a visit. And if there was a bigger gap, then it was a separate visit, if you see what I mean. So
yeah, brilliant. Thanks very much, Sarah. Was there much evidence of accidental spillage and take up by non-target animals or by disturbance?
No, so as I mentioned, the bait we used was hazelnut paste, so it's kind of a very thick liquid. When we added the bait dye, it was actually quite pink, so it was easy to spot. So every time we went to bait the feeders, we would check around them for any evidence of spillage. And we got very, very low levels of spillage because the squirrels would typically eat the bait in the feeder or just at the feeder entrance, and then they would kind of eat it all up anyway.
And it's not really something solid that they can take out and move around.
No, no, that's why we avoid solid food.
Yeah, that's brilliant. Back to here. Will you be considering smart traps that can send SMS messages when triggered, thereby reducing effort needed to check them? I think that might be one for Simon.
Well, it's perhaps one for Sarah. I don't know how far advanced those kind of traps are, but obviously, if we were to move to a different regime, a different approach where it wasn't necessary. So, for instance, the good nature traps, it's not necessary to check them daily anymore. Then we would obviously adjust our estimates and our for labour in our model, or we would try. And then build that into the comparison once we have a better estimate of how regularly traps are having to be checked. I mean, it obviously depends on in terms of the savings. Just thinking about it now, it will obviously depend on the numbers of squirrels you have, and I would expect at the beginning, if you've got quite a big population, it probably wouldn't necessarily save you much effort. But towards the tail end, as your effort or your success rate is lowered, then yeah, there's the potential that could mean you could trap out a woodland with less effort than we're estimating now.
Okay, brilliant. So something else to maybe incorporate in the future then.
Sorry, could I just add something?
Yes.
Sorry, it's just we do use smart traps at APHA. We use the ones that are based on vibrations. So when the trap goes off and vibrates, it pings an SMS to our mobiles. The only thing is that you need to be 100% certain that those trap monitors are working correctly, and you're not going to miss individuals in traps, obviously. So I think as a standard, we do go to every trap and check them just to be certain, because anything that's based on vibration could potentially have some error. I know some are being developed now which are actually small cameras focused on the trap, so you can actually visibly check them. And I think those would probably provide a more accurate kind of monitoring system going forward.
Thank you, Sarah. Have autonomous AI monitors to ensure delivery of bait only to greys been considered? One to you again.
So we're currently working with an AI company that has developed an AI model to distinguish grey and red squirrels from photographs. So we're actually using that as part of our grey squirrel density method to go through the thousands and thousands of photographs that we get from our wildlife cameras when we put them out to measure the density. However, there's some limitations with the AI. Firstly, would be the amount of energy or battery power that would be required to attach an AI element to each trap or feeder in the field. The AI at the moment, although it's very good at assessing photographs, it's not so great at low light. So particularly distinguishing between reds and greys, obviously a big element of that is the colour. And when you have infrared shots, which are kind of black and white, then it's actually really difficult sometimes for a person to distinguish whether it's a red or grey, depending on the angle it is facing the camera and stuff like that. So I think there's still a lot more development that is required before AI could be used cost effectively in the field. And I think our data showed that body weight could actually be a more cost effective way of distinguishing between the two species.
Yeah, thanks very much, Sarah. There's a question around, is there any problem to the public if they eat any grey squirrels that are caught for human consumption, or if bait were maliciously fed to a fertile human, would it be capable of causing temporary infertility? It's one for Bex.
So the first question, the answer is sort of the same to any predators or anything that would eat a grey squirrel. There shouldn't be anything left in the squirrel that would cause an impact on a human if it would eat a treated squirrel. But obviously, we will be going through the registration process and testing it in other animals. We wouldn't test it in humans. But yeah, we will be doing that safety testing. It's possible that malicious use could have some effect, but at the dose levels you would likely need for a human, it's very unlikely that it would have any actual effect on humans compared to that in squirrels, given the kind of differences in body size. But with the registration, we will also be looking at kind of licensing and restriction. And with the hoppers itself, we can look at locking them. So there would be reduce as much risk as possible, any kind of malicious use of this kind of thing.
Brilliant, thanks, Bex. If there were pine martens in the area where the bait was being deployed, will there be a way of keeping them out? Back to you, Sarah. I know you've talked about non-target species a lot, but
no, I know pine martens are one of the things that we certainly need to do more development in terms of the feeders. So in most areas where we'd want to deploy contraceptive, currently there aren't pine martens. So I guess we've kind of focused mainly on the common wildlife species that are likely to try and access feeders. However, we are aware that pine martens could be at risk potentially. So we could develop our body weight feeder to have a maximum threshold, as well as obviously pine martens are a fair bit heavier than squirrels. But we'd have to look at maybe redesigning the feeder slightly in areas where there's pine martens present, and we'd have to obviously thoroughly test that as well.
And you did do a bit of a study in Wales on looking at well, squirrels and pine martens, and with the basic feeder.
We did. So just as a little look-see, we put a couple of our feeders out in a wood where there was 2 pine martens, and it took them a couple of weeks before they actually managed to get into the feed, and they only did that a couple of times, but obviously a couple of times is too many times. But it kind of suggested that they certainly don't use the feeders as readily as grey squirrels do. So but yeah, we really need something a little bit more sophisticated to try and keep them out of our feeders.
Yeah, brilliant, thank you. Maybe on for Simon, do you envisage an eventual combination of approaches to reducing grey squirrel numbers, including culling, contraception, and other methods? And if so, how would these be integrated?
Yeah, so I think that's a good question, and yes, I think is the answer, that when we start to look at landscape scale control, from the point of view of even differing attitudes of people, we are going to be looking at an integrated solution with lots of different approaches in combination. Exactly how those are going to work together, you know, as we go along, hopefully if we achieve some of the additions to the model, like I said, including shooting, potentially including the impact of pine marten and looking at use of good nature traps, among other things, then we'll be able to start to inform those strategies and how things might work together. It's quite a complicated landscape, quite a complicated picture, to be able to say without trialing it, and even to be able to test it in a way, because there's so many different combinations of things that might be possible. But we've already shown through the work we've done that integrating 2 different methods, so the benefits of live trapping, for instance, to reduce a population quickly, then followed by the lower effort control from that contraception might offer, could produce a better solution together than just either contraception or live trapping on its own. So yes, I suspect an integrated solution will probably be part of the conversation, and probably work out to be one of the optimums.
Yeah, thanks very much, Simon. There have been a couple of people picking up on gene editing. I mean, I think at this stage, we all agree that it's early days. We just don't know what will happen over the next 10 years with that. But we will definitely keep our eye on it. And I know, Bex, you said that we'll be looking for opportunities to collaborate as and when that research moves forward. But yes, at the moment, we just can't say with any definite where that is really. I think that's let's have a look. Do you think there could be a negative chemical interaction between the hazelnut paste and the contraceptive itself that might reduce its effectiveness?
We don't know yet. So what we're doing is trying to work on developing a number of different bait types and working out which one is the most effective. We're trying a lot of different things that are hopefully as attractive to squirrels as the hazelnut paste, in case we get to the point where the hazelnut paste wouldn't work. But we hope that we can do a few things to the actual vaccine itself. There's components that we can add in that would protect the protein when we put it into different baits. So hopefully it can be as effective as possible in anything we put it in.
Brilliant, thanks. And is there any immunity from the contraceptive bait?
I don't know if that's getting into the realms of sort of, when poisons aren't effective on some of the population. We're not sure yet on the vaccine, so we would hope that as this kind of the first time that squirrels encounter this, there wouldn't be any immunity. We're trying to stimulate their immune system to it, but that's something that we're going to have to track over kind of large scale trials. At the moment in the lab, we're seeing kind of quite high levels of effectiveness across a whole group, but that's something that we want to be tracking and going forward.
Yeah, thanks, Bex. And I as far as I understand, the contraceptives that are used elsewhere in the world for other wildlife species, there's been no evidence of immunity as yet.
Yeah, definitely.
Thank you very much. In terms of future funding, are you wholly dependent on DEFRA, or you're approaching the forestry industry or major charities for funding? The UK Squirrel Accord is made up of 45 different signatories, many of whom have contributed to this current element of the research, and so have many individuals, as private estates, organisations, grant-making bodies, DEFRA, etc. Lots and lots of people have provided funding for the research to date, and going forward, we'll be asking everybody and anybody to contribute funding for this extension of the research and the next steps and the landscape scale trials. And we'll start our fundraising campaign off in the next couple of months. But yeah, just a huge thank you to everybody that has donated so far. We really appreciate it. And I think they're all of the questions. So I just want to take this opportunity to thank you and the rest of the APHA team involved for your hard work. I know lots of people want this today, tomorrow, but I just want to reiterate this is a novel and extremely complex project, and we can definitely celebrate the progress that's been made and look forward to the next steps to make grey squirrel control a reality. And I think that's a good point to wrap up. Unless anybody else from the team has any last words? No?
Just thank you to all the volunteers that contributed. That's all I'd like to say. I know I've seen some of them are actually on the webinar, so it's great to see them there, and just thank you.
Yeah, definitely. I mean, we've had a lot of collaborators, but the volunteers and having the volunteers involved has been fantastic, because they really are some of the powerhouses of red squirrel conservation out there. So thank you to everyone for joining us. I hope you found this update useful and continue to support our work. Our next webinar is on Tuesday, the 21st of May during Invasive Species Week. Emma Sheehy of Saving Scotland's Red Squirrels will be talking about the Aberdeen eradication efforts and the progress made there to enable red squirrels to return to the city. But for now, enjoy the rest of Plant Health Week, and I'll see you soon. Bye.